Good afternoon, everyone, and welcome to the second day of the Liver Seminar Summer Edition. Today, we will have two talks of our international invited speakers, and we will start with Dr. Gyongyi Szabo. She has accepted to join us, so we are very happy to have her here. She is the Chief Academic Officer of the Beth Israel Medical Hospital in Boston. She has a very extensive CV with a lot of publications and a huge and successful research group on immune response in the liver and steatohepatitis. She specifically asked me not to give many details on her biography to make it so extensive, but I invite you to revise her biography because it's so inspirational. So we are so happy to have you here talking about the mechanisms and clinical implications of inflammation in steatohepatitis. Gyongyi Szabo, please.
Thank you, Olita, and the organizers. I'm so honored to be here and giving this seminar, and good afternoon, everyone. And good whatever time of the day you have out on the internet there. So I'm going to talk about some of our recent studies, and unlike the really elegant presentations yesterday about all the new technologies, I think I'm going to be a little more mechanistic in a way of thinking about how liver disease sort of affects patients. So I thought it's better to start with my disclosures that pretty much have nothing to do with this particular talk, but the people who are depicted in my lab picture are, because they are the ones who actually really work together with me and have a fantastic group and privileged to work with them. So today I'm going to talk about a little bit of the clinical significance of steatohepatitis, and particularly focusing on alcohol-associated liver disease, MET-ALD, which is kind of a new category of the overlap between MASH and alcohol use, and a little bit about alcohol-induced acute and chronic liver failure. In terms of the mechanisms, I want to just highlight some of the roles of inflammation, particularly talking about some of the new discoveries that we have related to the BTK, the Bruton tyrosine kinase phosphorylation, neutrophil leukocytes, and particularly neutrophil heterogeneity, and the role of inflammasome activation. And along this, I'm going to show some of the potential therapeutic targets that we identified in this studies. So I'm not going to spend too much time on this, but you probably are very aware of that steatotic liver disease is becoming the most common type of liver disease globally. And that can be caused either by the metabolic dysfunction associated steatotic liver disease or by alcohol. And as I'm going to show you, or potentially the combination of the two. And you very well know that in MASH and MES-LD, there are multiple risk factors and genetic factors, as well as metabolic factors and diet, lifestyle, and so forth that contribute to all of this. Whereas alcohol use is typically a matter of time and demand, and potentially some gender differences in terms of women being more kind of having a lower threshold for developing alcohol-associated liver disease in the same amount of alcohol use compared to men. So I think in reflection of all of this, the joint statement from both the ASLD and EASL came up with this new category of steatotic liver disease. And particularly, I'm going to focus on this middle block here, which is the MES-LD and increased alcohol intake, which essentially goes from the spectrum of MES-LD to all the way to the separate entity of alcohol-associated hepatitis. But notice the fact that there could be either MES-LD predominant or alcoholic liver disease predominant kind of version of this MET-ALD, depending on the weekly alcohol intake. And the categories are, or the cutoffs for this are a little bit different between males and females, recognizing again this gender difference. This was defined based on identifying a drink as 14 grams per alcohol, which is the amount of alcohol in the US per drink. I think in Europe it's a little lower, so the numbers may need to change there. But nonetheless, I think when we step back and try to figure out what is the difference or potentially what are the common factors between these two diseases, there are many, many common factors. Fatty acid, the presence of free fatty acids is one of the common factors both in the MES-LD and alcoholic liver disease. Gene polymorphisms, the same PNPLA3, TMS2, and MBOT7 have been identified as components of the genetic kind of predisposition. changes the microbiome, gut dysbiosis. Both of these have been identified in both of the diseases. And of course, reactive oxygen species generation is the key component in some of the cellular injuries there. So today I'm going to talk about some of the common elements of the innate immune response. And I must say, when it comes to the combination of the MASLD and alcoholic liver disease, we primarily were interested in finding differences. And I'm going to show you that probably there are more commonalities than differences at the end. So probably don't need to spend too much time on this into the clinical progression of steatotic liver disease. You are very well aware of that the progression is in a way that during this process, in terms of the pathophysiology, multiple cell types contribute and actually crosstalk to one another. That amplifies the entire progression of the disease, both from the terms of fibrosis and also from the standpoint of inflammation that contributes to the progression of the disease. So let's start talking about a little bit alcohol. Alcohol affects multiple organs. We think about as the liver as hepatologists, but very well know that the alcohol has just as much effect on the gut. And in fact, as early as on the liver, because even a binge drinking can change multiple things in the gut, and particularly in the proximal small bowels. But chronic alcohol use has been shown to change the composition of the microbiome, to increase the permeability of the intestinal wall by impaired tight junction protein expression and triggering some inflammation in the intestine itself, and also increasing the potential translocation of microbial components such as pathogen-associated molecular patterns into the liver through the portal circulation. And at the same time, alcohol that goes into the liver, they can directly damage hepatocytes, particularly in a large amount of alcohol, because the metabolizing enzymes produce reactive oxygen species and essentially can damage hepatocytes. And this kind of brings in a perfect storm of both damage-associated molecules coming from damaged hepatocytes and pathogen-associated molecules coming from the gut to really fuel inflammation and activate inflammatory cells such as monocytes, macrophages, and neutrophils. All of this happens not only just in the liver, because systemically, particularly in alcohol-associated hepatitis, there is a lot of recruitment of these myeloid cells from the bone marrow that I'm going to show you that happens in this disease. I also want to point out that there is also alcohol can induce inflammation in the brain in terms of neuroinflammation. It has been shown in microglia, and recently we find it in astrocytes as well. And some of the molecular mechanisms that we find in the liver actually are very similar happening in the brain when it comes to innate immune cell activation. Most recent studies also suggest that potentially the addiction component of alcohol use is related to some of the inflammation that's happening in the brain. So that's kind of an interesting aspect. So I told you that both the pathogen-associated molecular patterns and damage-associated molecular patterns can activate cells. And all of this happens through evolutionary conserved receptor systems, toll-like receptors, and inflammasomes. That inflammasomes are intracellular, and the toll-like receptors, some of them are extracellular, some are intracellular. And I'm not going to go into details of that, but I particularly wanted to talk about a little bit about the NLRP3 inflammasome. That's an internal sensor of multiple cellular stresses, such as potassium efflux, reactivation species, some of the oxidized LDL or uric acid crystals can activate NLRP3. And this is a multi-protein complex in a way that it has a sensor component, which is an NLRP3, and has an adapter molecule called ASC, and an effector, caspase, which is caspase-1. And the importance of this, the caspase-1 is the effector molecule to cleave the pro-IL-1 beta into IL-1 for release. And IL-1 really has a role of kind of amplifying inflammation, as opposed to, for example, TNF, IL-6, or IL-E that are directly induced through NF-kappa B and immediately released. So turns out that this, on the right-hand side with the orange block, this NLRP3 ASC and caspase-1 actually can stay together and can get out of the cells, and it's called ASPEX. And we were interested in the role of this. But before I show that to you, I also wanted to show some earlier studies. We found that in alcohol-associated hepatitis, there is activation of this inflammasome. So we find that components of the inflammasome IL-1 beta, which is the kind of end product, serum and in the liver levels are increased, and also the components of the NLRP3 complex, pro-caspase-1 ESC and NLRP3 are increased caspase-1 activity. So this triggered us to look into the components of this and ask the question whether by deleting any of these molecules we could attenuate alcohol-associated liver disease. And in a mouse model, we find that indeed, if we use ESC knockouts or caspase-1 knockout mice, we actually find a reduction of all of these alcohol-induced liver disease components. And I'm sorry, I'm not sure what's happening up there. That kind of is totally scrambled. But IL-1 beta is reduced also. So because this NLRP3, ESC, and procaspase-1 complex can get out of the cells, these are called actually SPECs. And in some other conditions, has been shown that these SPECs actually get into the circulation, and they can be taken up by other cells and activate IL-1 and actually cleave pro-IL-1 beta in other cells. So this is kind of a way of maintaining inflammation potentially. So that kind of led us to the question that what happens in alcohol-associated hepatitis, where we know that patients stop drinking alcohol, come to the hospital because they're very sick, have this major inflammatory activation, but what sustains this inflammation? So the question was, could these aspects potentially be a reason for that? Because at that point, there is no more alcohol in the system. And in fact, when we started to look at these aspects, we find that using flow cytometry, that we find an increase in these aspects in alcoholic hepatitis in a human plasma, whereas in a healthy individuals, there is very little amount. And then furthermore, we find that these aspects actually are functionally active, because if we inject aspects into a naive mouse, then we find that there is actually inflammation induced in the liver, and we also find IL-1 in the circulation. So this is something that potentially physiologically could maintain and sustain inflammation. I'm not sure that this is the only reason, and particularly it's unlikely to be the reason for the multiple kind of month-long of inflammation in this patient, but certainly is one of the components. Furthermore, we asked the question that what happens if we inhibit this inflammasome and if we use an NLRP3 inhibitor, and here we use the small molecule inhibitor MCC950 that inhibits NLRP3 activity, and we find that indeed if we add the MCC950 together with the aspects to mice, then there is less amount of newly generated aspects, suggesting that this mechanism is indeed NLRP3 mediated. So we believe that these alcohol-induced aspects actually play a role. And the interesting thing is particularly binge drinking is the one that induces this aspect production. And we find that these intracellular aspects are present in alcohol-associated liver disease, but particularly in alcohol-associated hepatitis, or after binge drinking, these get out of the cells and get into the circulation and potentially can exacerbate the ongoing inflammation and be essentially a kind of perpetual inflammation that happens in this case. So we know that alcohol-associated hepatitis is a very poor prognosis clinically. Those patients that have a MAD score of greater than 20 are the ones that we start treating with corticosteroids because they have severe alcoholic hepatitis, but we also know that corticosteroids are not necessarily very effective. Furthermore, these patients are very susceptible to not only high levels of inflammation, but also to infections. And in fact, when patients get infected, very often they develop the systemic inflammatory response syndrome, go into organ failure, or if they had an underlying cirrhosis, they can develop acute and chronic liver failure. And at that point, they can have a very poor mortality or potentially rescued with liver transplantation. So we know that some of the kind of unique features of alcohol-associated hepatitis compared to many other liver diseases is the abundance of neutrophils in the liver. And neutrophils normally should play a major role in defense against inflammation, in defense against infections. So this kind of doesn't add up in the picture. So we were particularly interested in what could be the role of neutrophils. And in fact, neutrophils have multiple roles. They play a major role in acute inflammation in the liver, just as much as in any kind of other organ systems. They produce reactive oxygen species, phagocytose, dose degranulation, and also produce these extracellular traps that called neutrophil extracellular traps. But they also play a role in tissue repair and resolution of inflammation by removing debris and also programming potentially macrophages to become a more kind of anti-inflammatory macrophage. And the neutrophils that sort of perform their host defense job, then they get eaten up by macrophages called efferocytosis. So we were interested in kind of sorting out these roles in alcoholic hepatitis. And what we found was that when we did just this bulk RNA sequences between patients, alcoholic-associated liver disease patients and healthy subjects, that we found that there was a major difference in certain clusters of genes, particularly those that are involved in inflammation, which is not really that much of surprising. And at the same time, we also asked that what could be the mechanism for this. So one of the genes that showed up very highly in these patients' neutrophils was the Bruton tyrosine kinase, which is a non-receptor tyrosine kinase that plays a central role in mediating inflammation and in multiple cell types, including neutrophils, macrophages, and also potentially playing a role in steatosis. So we tested patients with alcohol-associated hepatitis. Then we also tested mouse and human livers with alcoholic liver disease or alcohol treatment in vitro. And we found in all of those conditions, BTK phosphorylation was increased, and particularly the BTK phosphorylation of both the serine 551 and the 223 levels essentially were upregulated, suggesting that this is really a very functional increase in the BTK phosphorylation. So that kind of led to the question that what happens if we actually inhibit the BTK, and this is something that you can do mostly in the animal model, although there are BTK inhibitors in clinical trials in myeloid diseases in patients, so the BTK is being clinically evaluated in other conditions in patients. But in the mouse, what we found is that if we give the BTK inhibitor, and those are the red columns, and either in a pair-fed or an alcohol-fed mice, this was a 10-day alcohol feeding followed by a alcohol binge. We found that there is protection from liver damage, there's a reduction of neutrophils in the circulation, and there is a reduction in the neutrophils in the liver. And of course, as we would expect, the BTK phosphorylation is also reduced in the presence of the BTK inhibitor. And this was true in the animal model, but also if we took neutrophils from patients with alcoholic liver disease and gave them a BTK inhibitor, that also inhibited the activation of these neutrophils. So then we asked the question that, are these neutrophils just happen to be in the liver, and obviously they will have to come from somewhere, and the bone marrow is the place where granulopoiesis happens. So then we evaluated the bone marrow in the mouse model and found that there is an increase in granulopoiesis in the model of alcohol-associated liver disease. There's increased number of neutrophils, and a number of neutrophils in the bone marrow, as well as their activation state, can be actually attenuated in the presence of BTK inhibitor, and also these granulopoiesis, early granulocytes actually show upregulation of the BTK phosphorylation in the presence of alcohol. So BTK is a kinase, so we were interested what could be the substrate for this, and if there are any specific substrate that happens in alcohol-associated liver disease. And doing a mass spec, we identified CD84, which is also called SLAM5, as a novel kinase substrate for BTK, and we were able to show that with co-immunoprecipitation. And in fact, if you look at CD84 expression in the patients, we found that there is an increase both in the human neutrophils and also in neutrophils from mice with alcohol-associated liver disease, that this is one of the molecules. So interestingly, CD84 has also been identified as a marker of myeloid-derived suppressor cells, suggesting that potentially these neutrophils that have the increased CD84 expression could have some inhibitory effect on immune responses, and that's something that we are actually evaluating. But some of the early signals supporting that hypothesis is the fact that if we take primary human neutrophils in panel G, you can see that if we use an anti-CD4 blocking antibody, then we actually can inhibit IL-1 production and also TNF production in these neutrophils, suggesting that this BTK and the CD84 interaction potentially plays a functional role in this activation process. So to wrap it up on this BTK story, what we find is that alcohol, particularly binge, activates BTK, and this is a very upstream sort of activity, and we believe that actually this is happening somewhere through TLR4 involvement. And as a result of that, there is increased granular poiesis, there's increased CD84, number of circulating and liver neutrophils and activation increases. But if we can inhibit all of this, then essentially this can be attenuated. So we kind of were trying to think about potentially clinical application of this. The caveat is that BTK inhibitors used in myelopoietic diseases in patients in clinical trials have a side effect of some liver damage, so that kind of puts some questions into this potential approach, but we are still evaluating that. So at the cellular level, we find that the BTK activation actually increases steatosis, which is a mechanism that we haven't fully explored. And I'm going to show you that also the BTK activation induces the net formation in the neutrophils, and this is happening through NLRP3 inflammasome activation. So what are the neutrophil extracellular traps? These are essentially the neutrophils. It's a host defense mechanism that originally believed to play a role in capturing bacteria or other kind of danger signals in the tissue environment. And neutrophils spill out decondensed DNA, histones, granular proteins, and neutrophil elastase. Those are the components of the neutrophil nets. So in alcoholic liver disease, if you look at this, then we find that actually there's an evidence for increased net production even in the serum of these patients. So it happens in vivo without a particular additional trigger. There is also increased cell-free DNA in the plasma, and also neutrophil elastase is increased in the serum of these patients. But when we take neutrophils from patients versus a normal individual, healthy, and then we induce net induction, then the patients actually show an exhausted phenotype. So they produce here on the right-hand side, they produce less nets than the equivalent amount of neutrophils from a healthy individual, suggesting again that something is already wrong with these neutrophils in the patients prior to isolation. So that led us to look into the neutrophil function a little more. And my graduate student actually did a bulk RNA sequencing in high-density neutrophils. But in the meantime, we also discovered that there is another population of neutrophils. And normally when you do like a fical isolation of neutrophils, the neutrophils are heavy because of the granules and come down with the red blood cells. And in the fical, the mononuclear cells stay on the top. But it turns out that in alcoholic hepatitis, there's a whole bunch of neutrophils in this mononuclear cell population. And this represents a different kind of a subset of, or a phenotype of the neutrophils, if you will. And these are low-density neutrophils. And the RNA sequences show that their RNA kind of pattern is very different. So the low-density neutrophils show a lot of downregulated genes in alcoholic hepatitis compared to either the high-density neutrophils from the same patient or compared to normal controls, whereas the high-density neutrophils in the patients show a highly upregulated profile compared to the healthy controls neutrophils. And indeed, the low-density neutrophils are very, very low abundance in a healthy individual to the point that we can't even isolate enough for functional assays. So this led to the question that what are these low-density neutrophils? And so we discovered that these low-density neutrophils actually are induced by alcohol in a way that in vitro, if you give alcohol to these high-density neutrophils in normal, healthy individual, then they essentially spill out the neutrophil extracellular traps. And here you capture the blue is the DAPI staining that shows in a second, the 1 hour picture, how the kind of the DNA is kind of going out from these cells, but then the neutrophils actually wrap up their cell membrane and continue to be viable. And on the right side, I'm showing that there is these actually after the net release, the neutrophils are viable. So essentially, this means that alcohol induces the viable and survival net formation, and the survived neutrophils become the low-density neutrophils. And these are the ones that have the exhausted defective phenotype that mostly we suspect that this contributes potentially a higher susceptibility infection. Whereas the high-density neutrophils are very active and this can sustain inflammation. And in fact, potentially this could be another way of thinking about therapeutic interventions. The other component of neutrophils is that came up in the previous discussions today that these neutrophils actually stay in the liver. So turns out that the alcoholic hepatitis patients, high-density neutrophils and also the low-density neutrophils have reduced expression of the CXCR4, which is the homing receptor expression for the neutrophils. So normally the the neutrophils after activation go back to the bone marrow to die. And because there is low CXCR4 expression, they stay in the liver. Furthermore, they also have a low expression of this signal, which is called CD47, that is also called as a don't eat me signal, which is the signal for macrophages, because normally macrophages would remove the neutrophils. So they have the don't eat me signal and then have a reduction in the eat me signal, which is the phosphatidylserine. So they essentially stay in the liver. And on the top of it, alcohol can also inhibit the phagocytic activity of the macrophages, thereby contributing to this hanging around the neutrophils in the liver. So then the next question comes that what do these neutrophils do in the liver if they're just kind of in the liver, and is it good or bad? Well, we have several evidence for how they are actually active in a way that if we take these neutrophils and induce the extracellular traps, then these extracellular traps can modify the function of, for example, stellate cells. So stellate cells is the one that produce collagen and contribute to fibrosis. So what I'm showing here is that if we use alcohol-induced nets, or a PA net means palmitic acid-induced nets, so this is a little bit relevant now to also metabolic dysfunction-associated liver disease. These nets induce activation of LX2 cells, which is a human cell line for stellate cells. And we can also kind of reproduce it. We try to see that which component of the net is causing all of this. So if we used citrullinated histone alone or DNA alone, that didn't induce the stellate cell activation. But when we added the two together, which is present in the nets, then that actually induced the activation of the stellate cells. And finally, if we disrupt the DNA using DNase on these nets, then we can essentially attenuate the activation of the stellate cells, suggesting that these nets actually, even in the absence of neutrophils, can activate the stellate cells. And they not only affect on stellate cells, but they also can change the activity of monocytes. So here I'm showing that the nets, again, these are cell-free nets, can induce activation of monocytes to produce IL-1 beta. But if we use a DNase, again, to disrupt the net, then we can prevent that. And then we found that using immunoprecipitation, that these nets actually interact with NLRP3, probably being a sensor for this. And if we inhibit NLRP3, then we can again inhibit the net-induced activation of macrophages. So to put this together, what we find multiple things about neutrophils, that alcohol-associated hepatitis has this kind of unique low-density neutrophils that are exhausted functionally, and most likely the result of previous net formation from a high-density neutrophil. High-density neutrophils are hyperactivated, and we also have evidence for the neutrophil infiltration being NLRP3 dependent. And importantly, these cell-free nets actually can promote fibrosis and inflammation, directly interacting with hepatic stellate cells or macrophages. In terms of therapeutic potential intervention, if we disrupt the nets using DNase, that's one way to attenuate it, or inhibiting NLRP3 with a small molecular inhibitor. Neutrophil depletion in an animal model is another way to show that we can reduce all of this, but obviously that's not something that we would want to do in a human situation. So we're kind of trying to put this together, but we believe that neutrophils could explain some of this immune paradox we see in alcohol-associated liver disease with really increased inflammation and the subclinical immunosuppression that happens in these patients. Certainly, we have to do more studies on this, but at this point, we believe that there might give a little bit of insight into the kind of unique role of neutrophils in alcohol-associated liver disease. So I'm going to switch gears now for a second and talk about the metabolic-associated liver disease. And you probably know that obesity sensitizes the liver to alcohol-associated liver disease. Depending on the amount of alcohol and the BMI, that there is certainly an additive effect here. And in fact, in our recent alcoholic hepatitis study, in a multi-center study, we find that the 90-day survival of those patients who had a high BMI, and that with the red lines actually is reduced both at 90 and 180 days. And this, interestingly, was associated with increased levels of multiple inflammatory cytokines in these patients. This is serum levels. And we also, if you look at the neutrophil activation in patients with alcohol-associated liver disease, that those have a high BMI greater than 25 have a higher level of CXCL1, lipocalin, or neutrophil elastase expression. So in a mouse model, we were interested that how can we combine the metabolic liver disease and alcohol and develop this MET-ALD model where we used a high-fat, high-cholesterol, high-sucrose diet supplemented with weekly alcohol binges over a period of 12 weeks. And this was in C57 black 6 mice. And essentially found that this increases the body weight in these mice, increases liver injury, increases steatosis, and also increases fibrosis quite substantially. And then when we did transcriptomic analysis, we identified various clusters of genes, and many of them were overlapping, but there were a few that were different in the MET-ALD group compared to either alcohol or MASH alone. So I'm not going to have time to show all of this, but we believe that in the alcohol and MASH diet, we not only get the liver damage and increased bilirubin, but also get the steatosis, which is more than just either of those alone, more inflammation, but particularly the neutrophil seems to be, again, a factor that we didn't necessarily expect, but it is happening very clearly. And in fact, there is increase in EMT markers, definitely a major increase in fibrosis, and also hepatocyte proliferation markers. So it turns out that the alcohol binges in this model of MET-ALD increase the infiltration and activation of neutrophils, which probably not surprising based on the previous data that I showed. But we also identified that there is an interaction between these various cell types in a way that, that in hepatocytes, there is induction of chemokines that will recruit neutrophils to the liver, and these neutrophils then activate actually I showed you earlier the hepatic stellate cells, and can also activate the macrophages through these various mechanisms. Interestingly, or potentially not unexpectedly, we find that NLRP deficiency or inhibition of the NLRP3 can ameliorate the liver disease in this combined injury model. So on the left, I'm showing the inhibitor MCC950 effects, and on the right, the knockout, which essentially just shows the similar protection from both the liver damage and neutrophil infiltration. And again, if we deplete neutrophils or treat the mice in vivo now with the DNAs to ameliorate liver damage, we find that there is reduction in the NETs and also in fibrosis in the combined model. And this suggests that the similar kind of strategies that I showed you earlier, NLRP3 inhibition or NET disruption, actually can be beneficial in this animal model of MET-ALD. So finally, I just wanted to spend a few minutes on acute and chronic liver disease. And the reason for this is because we know that alcohol is one of the most common triggers for induction of ACLF. Now, very often in patients with alcohol-associated hepatitis, we don't necessarily know what is the status of their fibrosis. So very often when we say it's an acute alcoholic hepatitis, it might be ACLF. So there's a lot of overlap between the two, but not necessarily the same. But clinically, they can present very similarly. And ACLF, there is increased mortality, there is multi-organ failure, and high mortality. So we were interested, and actually this is a work of Marty Ortega, who did his PhD here with Jordi, that there's no animal model for it. So we tried to establish a mouse model to really to have the key features of acute decompensation, multiple organ failure, and potential mortality. So in this, we used a cirrhosis model induced by bile ligation for 28 days, and then followed by an alcohol binge. And it is interesting because we needed to bring out this model to 28 days. If we did the same alcohol binge at 14 days, it didn't induce ACLF. So we believe that clinically this is kind of similar to what happens in patients that they really need to have advanced fibrosis or cirrhosis. And in fact, our model recaptures the ACLF pathophysiology both in the liver, causing liver damage. So again, in the dark bar on the right side shows the bile ligation plus alcohol, so the ACLF component. The fibrosis doesn't change because there is already fibrosis with bile ligation, and there is a lot of inflammation in the liver. But there's also extrahepatic manifestation in the kidney. We find increase in the creatinine and BUN, and also some early features of hepatic encephalopathy in terms of ammonia and also IL-1 beta neuroinflammation. So Marty was interested in identifying potential cell death pathways that are involved in this. And what he found was that there was no apoptosis, but potentially there is also necroptosis and pyroptosis happening in this model. And in fact, he found evidence for pyroptosis. So gasdermin D, which is a marker of pyroptosis, and potentially this is not surprising because I just showed you how the IL-1 pathway and NLRP3 activation happens. So gasdermin D activation can happen through that pathway. We find that, but also find evidence of necroptosis in terms of increased RIP3 kinase activation in this ACLF model. So then the question was, what happens if we try to intervene? And we used the RIP3 kinase inhibitor, which is not specific for RIP3 because it was both RIP1 and 3 inhibition here for the last 4 days of this model before and overlapping with the alcohol triggering the ACLF. And essentially what we found was that the RIP3 inhibitor worked, but then at the same time there was also reduction in liver damage in ALT. There was some benefit on the survival of these mice. And also the RIP3 inhibition reduced liver fibrosis, inflammation, and also attenuated the kidney injury and also some of the neuroinflammation that we see in the brain. So in conclusion, we believe that ACLF is improved by inhibition of necroptosis, at least in the mice, by using this model. And we are excited that hopefully this model will give us a little more insight into what's happening in ACLF. So to wrap up what I showed you today, that steatohepatitis and the progression of that to cirrhosis is really somewhat, at least partially mediated by innate immune activation in inflammation. And we believe that they're accumulating damage-associated and pathogen-associated molecule signals that contribute to this acute and chronic inflammatory cell activation and sensitization that contributes to this, both in terms of increasing the inflammation, but potentially also causing immunosuppression. And the molecular drivers include NLRP3, some of these aspects. We believe that the BTK is particularly important in activation of neutrophils, but it may also contribute to macrophage activation. And I'm not going to talk about the emerging biomarkers, but as you can imagine, some of these could be considered as biomarkers. And finally, potential targets and interventions are those that I kind of described earlier, both with NLRP3 or BTK inhibition, potentially CD84 blocking, and in the ACLF, potential RIP3 kinase inhibition. So with this, I'm going to close, and particularly wanted to acknowledge my colleagues in the lab and funding, and NIH for funding that may or may not be there longer. Anyhow, thank you so much. We haven't done that in a way that we know that we can identify this population, but we haven't had so one of the things that we are trying to do is some single cell in the liver, particularly focusing on neutrophils, both in the circulation and the liver. It's kind of hard to know that or imagine that these neutrophils that are already kind of went through one major phenotypic change to become low-density neutrophils, then NAV would be able to do something so much more because they mostly lost most of their DNA. So I'm not sure how much regenerative capacity they would have in the tissue or anywhere else, by the way. I think it's more of what's coming from the bone marrow, and that's what we're trying to figure out, that is there anything happening to the bone marrow precursors that potentially alcohol can pre-program them to be more active and do all these sort of functions. Yes, so clearly we believe that alcohol can induce the nets in the high-density neutrophils, and then those neutrophils that spill out the nets then can become these low-density neutrophils to hang out in the liver, and then they are mature neutrophils, because that was one of the questions I didn't have time to show, but this is in the manuscript, that we asked the question that is this like just an early kind of maturation stage of the neutrophils that potentially be in the liver. And we find that no, these are mature neutrophils and just kind of survive longer. We haven't really looked at like senescence or any of these markers. That obviously comes up as a question. Wouldn't be surprising if that plays a role. Yeah, so it's multiple things. Well, so we haven't really looked at in the MASH model in the bone marrow, but we looked at like BTK activation, for example, just as a control between alcoholic liver disease or alcoholic hepatitis and MASH, both in the mouse models and human. And there is some, for example, BTK activation even in MASH. The extent is much lower than what you see in acute ALCAP, which is not surprising because the extent of all the neutrophil infiltration and/or the activation is much lower. So, just for the purpose of these studies, we mostly focus on alcohol because it's just easier to get those populations. We don't see the low-density neutrophils in NASH necessarily, in the circulation at least, but that doesn't mean that it couldn't happen in fact, there's a little bit of increase in the liver. What you're asking is that, are there any these kind of mini hepatitis kind of events going on in MASH? I can't tell the answer to that, but it kind of is very likely or possible because we know that, all those triggers that can activate, for example, even NLRP3 with oxidized LDL and a whole bunch of, other of these signals happen in these patients, right? And we know that there is an autobrewery syndrome in MASH. So alcohol is there in a lower concentration. So all of these mechanisms, I think, actually can happen in MASH as well. In terms of pre-programming, so there is a lot of evidence that metabolic dysfunction and some of the metabolic disarray essentially can induce pre-programming trained immunity, particularly in bone marrow and precursor cells through epigenetic modulation. So that's something that we have evidence for now in alcohol. I'm not showing that data, but we have very kind of interesting evidence for that, and we're submitting a paper on that. I don't know necessarily in MASH, but wouldn't be surprising. But I think the extent is probably much lower. So that's a great question. So we haven't really done that side-by-side analysis for neutrophils. In the MET-ALD model that I showed you, where we had alcohol, the MASH, or the combination of the two, we did the transcriptomic analysis in the liver, but we haven't done it in isolated neutrophils. So I'm not sure that's what we really said, that was immature neutrophils. So we were particularly very interested in that question because for example, in LPS-induced models or in very acute infection in healthy individuals, there is this phenomenon called emergency granulopoiesis. And that emergency granulopoiesis is by definition that early kind of immature neutrophils coming to the circulation. So we thought that maybe this is what alcohol is doing. But what we found was that it's not true, because the neutrophils are actually more like mature neutrophils in alcoholic liver disease. But we do find the low-density neutrophils in the circulation. So the same, that's in fact how we kind of discovered them, that they were in the circulation and they were isolated out from the PBMC, low-density that is above the FICOL when you do like a density gradient isolation of peripheral blood immune cells. But those are not immature. So we know that binge alcohol or larger amount of alcohol induce NETs. In terms of how much NETs are present and what is the dynamics of NET induction in MASH, we don't know that. In this combined model of MET-ALD and alcohol binges, or MET-ALD, we find that the alcohol clearly is the trigger for this neutrophil activation. There might be some baseline, but it's much, much lower compared to what happens in alcohol, with alcohol, or in the presence of alcohol. Yeah, so I think you're referring to the fact that at the time when Remicade, the TNF blocking antibody came in, then there was an early clinical trial run at the Mayo Clinic in the US that stopped after enrolling about less than 10 patients because there was so much infection in those patients. So that kind of highlights the fact that these patients are immunosuppressed and highly susceptible to infections. And as we know, TNF and the pro-inflammatory cytokines play a major role in fighting infections. So when we discovered all this NLRP3 activation and IL-1, then we became very interested in the concept that IL-1 potentially is more like a modulator and an amplifier of inflammation, and if we block IL-1, then maybe we can just dampen the extent of inflammation, but not completely obliterating the kind of inflammatory and host defense inflammation component. And that led us to this clinical trial that was with anakinra, which is an IL-1 receptor antagonist. And I should say that I didn't show that data that in the animal model, this was more than 10 years ago when we showed that in an animal model, if we use IL-1 receptor antagonist anakinra, that beautifully obliterates the alcohol-induced liver disease and inflammation. So what we found in the patients is that, and this was a clinical trial with 4 different centers around the United States, a multi-center clinical trial. We used anakinra combined with pentoxifylline, because in those days we thought that pentoxifylline actually is protective for hepatocytes, and use zinc with the idea that can stabilize some of the intestinal sort of leakiness, if you will. And then that was against steroid, standard dose of steroid. We didn't reach the optimal number for the clinical trial in terms of enrollment because the study was over. But what we found was that in the first 28 days, there was no difference in the survival between the 2 arms of the study, but there were no significant differences in the side effects either. So IL-1 was much, much safer. Now, anakinra is a short-acting IL-1 inhibition, short-acting drug, so I think the IL-1 inhibition was not very robust. What we found was that 180 days, so 6 months later, survival was a little better in the IL-1 receptor antagonist group, but wasn't significantly different. So it turns out that just around that time, then Novartis actually engaged with Mark Turks in the UK, and they did this canakinumab study, which is an IL-1 antibody, and that has a longer IL-1 inhibition effect. But their outcome was liver biopsy results, and on the liver biopsy they didn't find a specific kind of improvement, so that was stopped. But they didn't look at any of the markers that we did in terms of survival. So I think the jury is still out. Oh, and then there was one more study that was a larger multi-center study that was with anakinra and zinc only, and steroid. And that clinical trial was actually, that was in the US, and it was stopped. This was run throughout the COVID pandemic. And it was stopped because, for reasons that nobody understands, I think, what we found was that there was essentially no death and no mortality in the steroid group. And compared to that, the anakinra treatment seemed like there is more death, but the anakinra behaved exactly the same way as in the previous study or with previous steroid treatment. So that kind of was the end of the IL-1 receptor kind of treatment still, I think the jury is still out. No, we haven't done it that way. And I think, as I showed you, because again, this is the first time we tried that, we used 4 doses. So we used it for 4 days, or 3 days before alcohol. Alcohol was given, and then like a half an hour later we gave another. Partially because we didn't know what is the half-life of this inhibitor, but certainly that would be the next logical step to kind of see what is the effect if we give it after.
We had a great session, and then we will reconvene at 4:30 sharp. So thank you so much for your attention and your collaboration. Thank you.